What you've just seen may be an actual solution to managing invasive parasitic sea lamprey populations in the Great Lakes. Although fish aren't exactly dousing themselves in repelling odors, scientists have recently found that sea lampreys in the Great Lakes avoid odor or putrefaction extracts obtained from other decaying lampreys. These odor extracts may provide an affordable and sustainable method for managing invasive sea lampreys in the Great Lakes. Before diving into this study, let's first explore some general characteristics of lampreys. Lampreys belong to the clade Agnatha, a general group of vertebrates within the clade Chordata. Members of Chordata usually exhibit bilateral symmetry, segmentation, 3 germ layers, and a complete digestive system in a well-developed coelom. What sets this clade apart from others are 5 general characteristics present at some point of each of its members' life cycles. These characteristics include the presence of a notochord, pharyngeal gill slits, a hollow nerve cord on the dorsal side of the body, a postanal tail, and an endostyle or thyroid gland. Lampreys are no exception to these developmental features. Gill slits and a postanal tail are 2 of the most obvious characteristics, as they are easily seen on the adult lamprey body. Adult lampreys also possess a notochord and a hollow nerve cord in the form of a spinal cord on the dorsal end of the body. The endostyle is also present, but only in the larval stages of the lamprey's life. Sea lampreys are native to the Atlantic Ocean, but were accidentally introduced into the Great Lakes through ship canals and man-made locks in the 1800s. Due to an abundant fish stock in these lakes, they thrived. Sea lampreys are anadromous fish. During the spring and early summer, they ascend freshwater streams and rivers to spawn. The males and females seek out areas to create nests of small stones and gravel. Females may lay 30,000 to 100,000 eggs. After spawning, the adult sea lampreys die. In a few weeks, their eggs will hatch. The young larvae then leave the nest and drift downstream, where they burrow into the stream's bank and remain harmless larvae for 3 or more years. After this period, sea lampreys undergo a transformation phase in which they begin to develop eyes and suckers with sharp teeth. The sea lampreys then swim downstream to the Great Lakes, where they feed upon and parasitize the fish population. Once the sea lampreys have reached sexual maturity, they stop feeding and swim upstream in preparation for spawning. Lampreys have been shown to rely on pheromones to navigate into streams with suitable spawning and rearing habitats. Lampreys also rely heavily on pheromones during mating. Scientists in the past have relied on these odors to lure lampreys into traps. This, however, is not always effective. Instead of attracting lampreys away from a location, a study published in the Canadian Journal of Fisheries and Aquatic Sciences sought to keep lampreys out of areas altogether by manipulating odors released by decaying lampreys. These odors had previously been shown to act as alarm cues for lampreys, keeping them away from risky areas associated with predation, contagion, or death in general. If researchers could harness these odors without relying directly on decaying lampreys, perhaps a new and effective alternative could be used in managing invasive lampreys in the Great Lakes. To test this, 160 healthy adult migratory phase sea lampreys were obtained from the U.S. Fish and Wildlife Service. These lampreys were separated into 16 groups of 10 individuals. 8 of these groups, or replicates, were to be subjected to an ethanol control, whereas the other 8 were to be subjected to a putative alarm cue, which was extracted from individual lamprey carcasses and combined into a solvent containing ethyl alcohol and water. This figure shows an overhead view of the water tank racetrack in which the lampreys were observed. Trials consisted of 3 periods: a 20-minute pre-odor period, in which lampreys were released from a cage and allowed to swim freely, a 40-minute exposure period, where odor was released from pumps into one side of a channel, and a 40-minute post-exposure period, in which odor exposure ceased and lampreys swam without any odor cues. A video was recorded of each trial in which the locations of the lampreys relative to the 6-square grid shown in the figure were marked. Researchers found a significant difference between the behavioral responses of the lampreys in the presence of the ethanol control and the putative alarm cue. As shown in the figure here, lampreys exhibited no response to the ethanol control but actively avoided the stimulus side of the racetrack in the presence of the putative alarm cue. These findings ultimately show that sea lampreys can be controlled with use of a natural repellent. The authors proposed that this could be used to divert migrating lampreys from repellent-treated tributaries and trap them using a migratory pheromone. Ideally, this would greatly limit the amount of spawning area available to lampreys. This study ultimately presents 2 large opportunities. The first being that using a novel substance, in this case the predatory alarm cue, may intervene with reproductive decision-making of a migratory species. The second being that alarm cue extracts may lead to efficient pest management of lampreys. as the extracts are cheap and easy to obtain. With more research stemming from this study, perhaps we will gain better control of invasive sea lampreys in the Great Lakes.